Thank you. We are excited to get started today. I'm going to pass it over to Roseanne. She is our staff writer here at Hope and Focus, and she will take it away. Thank you.
Thanks, Courtney. Welcome to Let's Chat About. This is a free bi-monthly webinar series hosted by Hope and Focus. We've developed the series with those living with LCA and IRDs in mind, but it is open to anyone who is interested in what's happening in our communities. My name is Roseanne Smiley, and I'm the staff writer for Hope and Focus. Before we get started, I want to take a moment to thank our sponsors for this series: AGTC, Dominion Energy, Janssen, Mira GTX, ProQR, and Spark Therapeutics. We could not provide programs such as these without their support. A few housekeeping items. The session will last about 45 minutes, and we've turned off the chat function during the webinar. You can submit questions through the Q&A function, and we did collect questions in advance from the registrations, and we'll be going through those first. As time allows, we will take any questions that come in live. We are recording this session. After the webinar, we will post the recording to our YouTube channel and send you the link. It is my pleasure today to welcome Daniel de Boer, founder and chief executive of ProQR Therapeutics based in the Netherlands. Daniel is a serial entrepreneur and a passionate advocate for rare disease patients. After one of his children was diagnosed with a rare disease, he started ProQR to develop RNA therapies for rare diseases. Under Daniel's leadership, ProQR developed a platform that yielded a diversified pipeline of potential treatments for rare diseases and raised more than $400 million in funding, including an IPO on Nasdaq. Before founding ProQR, Daniel was founder and chief executive officer of several technology companies. Daniel is also co-founder and strategic advisor to Amylon Therapeutics and Wings Therapeutics. He's a strategic advisor at Frame Therapeutics, Meatable, Algramo, and a member of the advisory board at the Termeer Foundation. In 2018, Daniel was named Emerging Entrepreneur of the Year by EY. In 2019, Daniel was selected for the Young Global Leader Program at the World Economic Forum. Daniel, welcome, and thank you so much for taking the time to chat with us.
Thank you, Roseanne.
We're so happy to have you. I want to start with your personal story. You have a child with a rare disease, and that led you into pharmaceutical development and the founding of ProQR. Can you talk to us about your journey through rare diseases?
Yeah, happy to. But first of all, from all ProQRians, I want to congratulate you guys on at the Hope and Focus team on the rebranding that you did at the end of 21. I think it really shows the continued journey your organization is on and how far you've come. And I also want to say a big thank you to you, Roseanne, and also Alyssa before you, as well as the wider team at Hope and Focus for the invitation. I think for us, it's always a great honor to be invited to speak to our key community partners. So, Roseanne, you mentioned I'm Daniel de Boer. I'm the founder and the CEO at ProQR. And I founded this company back in 2012, which I indeed did when one of my children was born and diagnosed with a rare genetic disease. This was a few years before that. So when he was born, my son, I decided to see what I could do to accelerate drug development for rare diseases, which led to starting ProQR Therapeutics. So at ProQR, we are dedicated to developing life-changing RNA therapies for patients that suffer from genetic eye disorders like Leber's congenital amaurosis, Usher syndrome, retinitis pigmentosa, and a whole range of other diseases that we are working on. At ProQR, we are based essentially around the world, but you will find most of us in Leiden, the Netherlands, but also in Cambridge in Massachusetts. And today there's over 200 ProQRians representing more than 30 different nationalities. So we at ProQR are very motivated by our mission to help patients, and we do that by creating RNA therapies that aim to stop vision loss or even reverse some of the symptoms that inherited retinal diseases cause. And yeah, we know that LCA is the leading cause of childhood blindness at least in the Western Hemisphere. And that gives us at ProQR a tremendous amount of sense of urgency to work hard on accomplishing our goals. We see that there's a really large unmet medical need. So there's more than 5 million people in the world that have a form of an inherited retinal disease, and just very few of them have a treatment available to them. And at ProQR, our plan is to change that.
Okay, very good. You mentioned that your company focuses on RNA therapy. What's an RNA therapy?
Yeah, so an RNA therapy is a really innovative technology that treats genetic eye conditions such as LCA10 or Usher syndrome. And RNA is really important because the RNA helps to carry out the instructions that are in the DNA to make proteins. So we're all familiar with genes and DNA that we have in our cells. And the RNA is essentially helping to carry out the instructions that are described in the DNA, which is to make certain proteins. And these proteins are critical to a healthy, the healthy functioning of a cell. So in genetic diseases like LCA10, there is a mutation in a gene, and that mutation gets copied into the RNA, and therefore we cause a loss of protein. So you see that a certain protein is not functional or not there at all, which leads to a cell not being able to function well or even to die over time, as is the case in LCA10. And this is, for example, where we then see inherited retinal diseases develop, where it's really caused by the lack of these proteins. So RNA therapies can repair the RNA without altering or changing the DNA. So we don't have to touch the DNA. We don't have to change any of the genes. We can leave all of that untouched, and we can alter the RNA in between such that the cell can make its own functional and healthy proteins. So we're currently developing RNA therapies in clinical trials for a whole range of different diseases. So this obviously includes sepofarsen, which is our lead therapy, which we are developing for Leber's congenital amaurosis type 10, which is in a phase 2/3 pivotal trial that will read out later this quarter. So that's really exciting, really around the corner. And I'm sure we get to talk about that a bit more later on in today's session. Then our second program is called QR421A, which is for a disease called Usher syndrome and Usher associated retinitis pigmentosa. And for this program, we just started 2 parallel phase 2/3 pivotal trials that are also being executed globally. Then our third RNA therapy that's in the clinical trials is called QR1123, which is for a specific form of retinitis pigmentosa called autosomal dominant retinitis pigmentosa. And this program is in a phase 1/2 trial at the moment. And then our fourth program is called QR504A for Fuchs endothelial corneal dystrophy, which is also in a phase 1/2 trial. And then beyond that, we have several preclinical stage programs on other forms of IRD, so including LCA, Usher, retinitis pigmentosa, and Stargardt's. So RNA therapy can really be applied very broadly, and we're applying it to start with in these indications.
Wow, that's a lot of stuff you got going on in that pipeline. That's wonderful. You mentioned a little bit about RNA therapy and how it works. We're pretty accustomed now to say what Spark Therapeutics developed with Luxturna, with the subretinal injection into each eye. How do you administer this RNA therapy?
So RNA therapy is administered through intravitreal administration, and that's short is IVT. And that's a really common procedure that's really established for all kinds of different retinal diseases. So you may have heard of conditions like macular degeneration or AMD, where IVT administration is the standard of care, and any ophthalmologist can administer a drug through IVT administration. With that IVT administration, so intravitreal administration, which is a small injection in the side of the eye, in the wide part of the eye, which is a 15-minute procedure. Through that route of administration, we have a big advantage that we can treat the entire retina. So only with a small injection in the side of the eye, the RNA therapy will distribute itself throughout the entire eye and will go to all different parts of the retina. So that means that we can treat both the central retina as well as the peripheral retina, which allows us to, for example, also treat early stage disease, which generally starts in the outer part, in the peripheral part of the retina. So yeah, that's RNA therapy is administered through IVT administration.
Okay. Okay. If we, as you know, we're very interested in your LCA programs, including especially LCA10. But how did the LCA10, your CEP290 research begin? How did you get into the LCAs of things?
Yeah, so we started out at ProQR as a cystic fibrosis company. My son was born with cystic fibrosis, and at some point there was another company that developed a really good therapy for cystic fibrosis, which meant that what we were developing at the time was no longer necessary. Okay. And then in about 2018, we decided to completely move away from cystic fibrosis and essentially reinvented ourselves as an ophthalmology company. And that was not overnight. We had been working in the ophthalmology field since 2014. Actually, we had a partnership with Professor Rob Collin from the Radboud University in Nijmegen, which is in the Netherlands, where Professor Collin had discovered an RNA therapy for LCA10, so an RNA molecule that he had published on, which later evolved into becoming sepofarsen. So we had really been working on this for years and years before we were at the point of starting clinical trials. And sepofarsen then entered clinical trials in 2017 when we started our phase 1/2 trial. And in 2018, we did an interim analysis in that study, which was before the study was completed. We took an early look at the data, and we saw examples of some really transformational improvements in vision. So, for example, there was a participant in the trial that started out with light perception vision. So it meant this individual could only see day or night, but not any shape or motion or form or color. And after a single dose of sepofarsen, this participant then improved his vision such that he could now read, he could recognize people's faces, and he could essentially navigate the world independently for the first time in decades.
Wow.
Yeah. So with these results, we were obviously really excited for the impact that this showed that the RNA therapy could potentially make in IRDs. And we also saw our hypothesis confirmed that RNA therapy in the eye could potentially make a really meaningful impact. Wow. So then, fast forward to today, we completed our Illuminate phase 2/3 pivotal trial for sepofarsen recently and are now awaiting the results that are being analyzed. And we're obviously really excited to see the results of that trial later this quarter, which really is now around the corner. We hear a lot of different communications and webinars where research really seems to be far away, and it seems to be 5 to 10 years for getting something into the hands of the community. But with sepofarsen, the data readout is really just around the corner, which means that this year we could potentially see a second treatment for LCA this time for LCA10, following the Luxturna readout that we saw a few years ago. And that's exciting, and it shows that the innovation in the IRD space is coming and it's really accelerating. And we're very encouraged by that. So we remain on track as a company to publish our top-line results from the phase 2/3 pivotal trial, the Illuminate study, as it's called, later this quarter. And I know that you guys at Hope in Focus work closely with our patient advocacy lead, Andy Bolin, who will provide the community with a plain language briefing on what the data means once it's published, because these things can be very technical and complicated. So Andy will help to make sure everyone can interpret what the results of this trial show. And then I want to mention that we have some, I think, wonderful video and written resources outlining data and progress that we've made to date on our website. So these can be found on ProQR.com. And any questions that may result from that, people can address to our patient advocacy group, which can be reached at patientinfo@proqr.com.
Oh, that's very good. Thank you. Thank you for mentioning that. You talked about your Illuminate trial, and now we saw news that ProQR launched Brighton. It's a pediatric study in LCA10, but it's only in the EU. Why not in the USA?
Yeah, that's a great question. So LCA10 is a pediatric disease, right? It starts affecting children really early on in life. And ideally, you would start treating children as early as possible. We therefore have decided to include patients in our development program essentially from the get-go. So in the first trial that we ran, we included children of 6 years and older, and we got 1 or 2 pediatric participants in that trial, which is really important to learn about the effect of sepofarsen in this population. And then in the phase 2/3 pivotal trial, the Illuminate trial, we are including patients of 8 years and older. So again, a lot of pediatric participants across the development program. So that means that also once we get to the Illuminate results, which are later this quarter, we will also be looking at pediatric data from that trial. So that's encouraging. In addition to all of that work being done, we're also running the Brighton trial, which is studying in even younger children, mostly for safety, because the really young children, for example, cannot read the letters on the eye chart as they can't read yet. So being able to really measure for efficacy is more difficult. But given this is such a small trial, we only run that in select countries. But with the Illuminate trial being progressed, we hope that we can put sepofarsen into the hands of the community very quickly. And again, I would encourage people, families that have children with CEP290 mutations, to reach out directly to patientinfo@proqr.com if you have any additional questions on clinical trial participation or anything else.
Great. Just a follow-up, are children with low or no vision included in Brighton?
Yes, really good question. So in the Brighton trial, we are including patients that are light perception or better. So that means that the no light perception patients can unfortunately not participate, but the light perception patients can participate.
Okay. Okay. It's crucial to incorporate different people in clinical trials. And how is ProQR doing this?
Yeah, I think it's very important to incorporate people from across the world in our trials and from all different backgrounds. We also think it's really important to incorporate people that will ultimately use the product in the work that we do. So what we do is we execute our clinical trials across multiple countries. So I think the sepofarsen Illuminate trial is probably being conducted in 10 or 12 different countries in Europe, North America, South America, to really make it accessible for participants to participate in the trial. And then really importantly, we think it's crucial to integrate the patient voice into our decision making throughout all steps in the drug development process, as we believe that as a patient-focused company, that is really crucial to our success. So at ProQR, we do this in several different ways. So first of all, we really engage with families and individuals that are affected by any of the IRDs that we work on. And we have regular webinars, fireside chats, and presentations that we broadcast in the company. So we, for example, had Laura from Hope in Focus late in 2020 participating in one of these webcasts that we do in the company. Secondly, we really aim to listen and engage with groups like Hope in Focus and others. We think that the community has critical insights that can help us as a company to really shape all aspects of our work. So the clinical trial design, the regulatory discussions, quality of life measures, et cetera, et cetera. And to that extent, we have a dedicated team with patient advocacy lead Andy Boland that can help any questions that arise from that. We also formed a patient and caregiver steering committee, which is a group of people that we meet with on a regular basis to help us address all kinds of questions that can help us to develop the best possible medicines for people living with IRDs. They help us on clinical trial designs, on endpoints, on measures, but also really understanding quality of life impact. And I think that's crucial. It really helps us to be better informed as we go through our development programs.
Absolutely. You mentioned a few other rare IRD populations, like RP and Usher. Are any other IRDs that are in your?
Yeah, so actually, I think that is our ultimate objective, to really develop a diversified portfolio. So a lot of different medicines that each target very specifically a certain disease mutation. So that's throughout essentially a group of different medicines. We can treat a very significant group of people that live with inherited retinal diseases. So we have a deep pipeline to potentially accommodate that and to make that possible. I went through the 4 programs that are in the clinic, and beyond that, we have in preclinical stages several other programs that, for example, target other mutations in LCA, other mutations that lead to retinitis pigmentosa. So, for example, other Usher mutations, but also other mutations that lead to RP directly. But also Stargardt's is in our preclinical pipeline, and a number of other indications. Across the board, we are fully focused on genetic eye diseases. We're really specialized in that. And over the years to come, some of these programs that are currently earlier stage may progress forward into clinical trials as well. And we plan to keep people updated about that as we progress.
Wonderful. That's great. So now we're going to get into some of the submitted questions. Supply chain disruptions due to the pandemic have created lots of bottlenecks in manufacturing. Have you experienced any slowdowns because of any supply deficiency at any of the manufacturing stages of your product or in the quality control?
Yeah, that's a great question, Roseanne. Yes, I think supply chain has become more challenging. Fortunately, we've not been affected by that. Our manufacturing from start to finish is well under control. It's being done at several different locations. So we have fallback scenarios for manufacturing plants to fall back to if needed. So we have not encountered any issues, fortunately. Both not for the clinical trials as well as for the material that we would be preparing for the post-approval situation.
Oh, that's great. Okay. Let's see. Well, I think that's what I have for submitted questions. Do we have any questions from our? Let's see what we have here. Okay. Here's a question from a listener or a viewer. It says, assuming positive results soon, fingers crossed, are there any challenges you anticipate moving forward with sepofarsen? And is there anything that the patient community can be doing?
Well, what we do is not easy, so there's always challenges. But I don't see any particular challenges that we would not be able to mitigate or overcome. I think the patient community is really important in the process. I think the awareness of the disease, awareness of potential therapies that may be coming is crucial. And be able to really inform the community about it, I think, is of the utmost importance. So I think the community could really help by making sure information is disseminated. And I think Hope in Focus is playing a crucial role in that process as well.
Okay. So it clearly helps for you to hear from your patient communities. I can imagine. Let's see. We've got. I think there was another question, but let's see, where did it go? I think it was in the chat. Okay, let's see. Will you be applying for FDA approval I suspect this is of cepofarsen in the US and Europe simultaneously?
Yes, that's a good question. So we plan to make this drug available to patients wherever they are. So our strategy is indeed to file for registration in both Europe and the US in parallel, and then make the therapy available as soon as we get approval. We are not limiting ourselves to the US. We're also obviously active in Canada and in certain places in South America.
Okay.
So yeah.
Okay. Great. Let's see. I think there's another question here. Oh, yes. Yeah, it just says yes for cepofarsen. Okay, let's see. Another question. When do you think you will be applying?
Yeah, we haven't. That's a good question as well. We haven't given guidance on that because it really depends on so many factors, and it may change, and we don't want to kind of go back and forth on that timeline. But we'll certainly inform people once the data is available. Once we announce that, we will give some projection on when we plan to submit. Obviously, as I mentioned at the beginning, we feel a really strong sense of urgency to get this therapy into the hands of patients. So that's what we will be focused on.
What are some of the challenges to get from here to applying for FDA approval?
Well,
just
yeah, I guess
a few examples.
Yeah, I think we could dedicate a full webinar to talking about the regulatory pathway. But yeah, in short, the regulatory submission is a submission of a very extensive file that describes everything that is known about the potential therapy. So everything from the science that has been done in the lab, the manufacturing of the product, the quality control thereof, all the clinical trials that have been conducted, everything that has been touched by the product over the years that it has been tested. All of that needs to be written up in a diligent and controlled way, in a really extensive set of documentation, and that is then submitted to the regulators. You can imagine that there's a lot of work that goes into putting that file together, then having it submitted, but also at the side of the regulators to then read all of that, interpret it, and have an opinion about it. There then follows a period where there's a lot of interaction with the regulators to clarify any outstanding questions. After which the regulator will form an opinion and make a decision on approval. So yeah, there's quite a lot of wood to chop, but nothing that hasn't been done before. And yeah, we have a really experienced team that is really geared up and prepared to do this.
That's very exciting. Very exciting. Okay, let me see what else we got here. Okay. One of our viewers is saying, what is the current timeline for the use of cepofarsen? For instance, how long before it has to be repeated?
Yeah. So that's a really good question. So RNA therapies generally need to be administered twice a year. So 2 times a year, people take a dose of cepofarsen in each eye. So far, we have seen that leads to the best possible results, with a rapid increase in vision and then a sustained benefit over longer periods of time. So twice a year is the optimal dosing interval in the studies that we've done so far.
Okay, thank you. Let's see, how much does the population associated with a particular IRD affect your interest in developing a treatment?
Really good question. Obviously, the incidence and the prevalence is something we look at. We have to look at. Mostly because if the population is too small, it becomes really difficult to develop a drug. If there's not enough patients to really participate in a trial, yeah, we can do all the work we want, but we will not be able to conduct a clinical trial. LCA10 has probably around 1,000 patients in the US or in North America that have the P699X mutation. That is the most common mutation in LCA10. And that's considered one of the smaller inherited retinal diseases. So we're not too sensitive to small populations. I think we want to make sure that we really provide possibilities to all patients that have an IRD. But in all fairness, I think we're mostly driven by where the science leads us. So RNA therapies can be applied to certain types of genetic mutations. I won't go into the details here, but it's pretty well described on our website what our technology can do. And that really dictates what kind of mutations we can treat and what kind of mutations we cannot treat. So there is kind of a natural selection where the science guides us, and that's where we focus and prioritize.
Okay. Let's see. Another question is it too early to prognosticate about how much a treatment would cost?
Well, it's certainly something we're thinking about, but only once the pivotal data is available, you can really form an opinion about that, because that will tell us something about how effective the therapy is. And as we move forward, we will learn more about which patients can use the therapy. And all of that will help us to inform where the price should be to make the product commercially viable. And as we just discussed, Roseanne, there's 1,000 patients in the US or in North America that have this mutation. That's a relatively small group of people. So we will do the work over the coming year to land in a place where, in a responsible way, we can make this a commercially viable medicine.
Got it. Okay. And another attendee just wants you to go over again, if you could tell us the difference. They're talking about between an RNA medicine and a DNA medicine.
Yes, happy to. So in a cell, our body is made out of cells, right? Billions of cells. In a cell, there is DNA, which we're all familiar with. That is kind of the library of our genes. So this holds all the information that our cells need to function. So what happens in a cell, the DNA is copied into the RNA, and the RNA is essentially the blueprint that then makes proteins. And proteins are expressed in your cell to do all kinds of different tasks, and essentially is what makes our bodies function. Now, with RNA therapy, what we can do is we can repair the blueprint. So we give it an RNA therapy that repairs the blueprint, and from this repaired blueprint, the cell can now make its own new functional protein. So that's what an RNA therapy does. With a gene therapy, you essentially replace the gene, so put a gene into the DNA, which then expresses RNA that makes protein. So I think an advantage of RNA therapy here is that, first of all, we can do IVT administration. So that is a really simple route of administration that helps to treat the entire retina, which is really important, especially in early disease. So, for example, if you would want to treat kids, you want to be able to treat the full retina. And then for RNA therapy, you don't need a viral vector. So gene therapy often is packaged in a virus. So this is a virus that is made in a way that is no longer harmful to human, but it's used as a delivery mechanism. So this virus is then loaded with a new gene and injected into the back of the eye, where it then is entering the cells and is expressing the protein. With RNA therapy, we don't need to use any viruses. So we can just administer the therapy, so the RNA therapy, and it will find its own way into the right cells. So the difference is really where RNA is repairing the RNA such that the protein can be made. With DNA, and you actually have to change something in the genes to make a permanent change.
Right. Very good. Okay. Let's see. I think we have another question here. Okay. What have you learned from your trials so far, which can speed up the process from preclinical to approval? And they say, and which you have excelled at thus far.
Well, thank you first for that. As I mentioned in the beginning, I came from the IT business. I had a number of computer companies before starting ProQR. Getting into the pharmaceutical world, I really had to get used to these extremely long timelines of drug development and everything that's associated with that. So ever since the start of the company, which is almost 10 years ago, we've been very focused on finding efficiencies and accelerating drug development where that's possible in a responsible way. So one of the things we are doing in parallel to developing the therapies that we have in our pipeline is actually trying to find faster ways to develop medicines by being much better informed from the work that we can do in the lab. And for example, one of the things we are doing in the lab is growing human retinas. So we can, these are called organoids. We can take a skin sample from anyone in the world, but we're obviously mostly interested in the samples that come from people that live with an IRD. And we can, out of these skin samples, grow a human retina in the lab. And from this retina, we can then test the activity of our therapeutics. So we can administer drugs on these retinal organoids, which then tell us in the lab already if they're going to be functional, if the drug is going to work once we give it to a person. All of this is obviously in testing phase still, so we can't have 100% certainty that the preclinical model will always be predictive. But so far, we have seen that in both cepofarsen and in Usher, the model was really spot on in predicting the activity, but also the active dose level that we had to give once we started clinical trials. And if you think about that, Roseanne, I think there is really potential to find more synergies and really speed up the development from preclinical to approval once we generate some more data across more of these programs that can help us to validate the correlation with the preclinical models to potentially really accelerate the development timelines.
Wow. Yeah, that's wonderful. That whole retinal organoid is just fabulous. I mean, you don't have to put people at risk, really, Let's see. Oh, and we've got, okay, this is a good question then. Have you received any negative feedback from the FDA about using retinal organoids?
No. We actually were selected by the FDA for a program that they called model-informed drug development, which is essentially a program by the FDA to work with companies to see how you can use preclinical models to inform the drug development process. And so no, actually, we've not received any negative feedback. We've really collaborated with the FDA and with the regulator in Europe on further seeing what the potential is of this model. So yeah.
That's perfect. That's great. Okay. But you mentioned that you're collaborating with the FDA. That seems to be the same thing with the importance of the patient voice. It's something that's been that the FDA is also collaborating with companies like yours to make that central to your research. Okay.
Absolutely. No, I think obviously the FDA has a single purpose, which is to get effective and safe therapies approved and available for the population. And they share the same goal essentially as we have collectively as the community. So yeah, we really partner with the FDA and other regulators to make that happen.
Okay, let me see if I think any more questions. I don't have any more questions here. Anybody want to submit something? I don't see any. Daniel, would you like to add anything else? I think we're kind of winding down here. You've been a wealth of information and kept us updated on a lot of great things that you've got going in your pipeline. Anything else you'd like to say?
Well, if anyone has further questions, you can visit our website. We have a dedicated community website actually on ProQR.com, where there's guest blogs, there's interviews with families, there's plain language updates on all of the work that we do, both science as well as clinical trials. So you can find that section on our website at ProQR.com. If there's any more questions, you can reach out to patientinfo@proqr.com, where our patient community group can help you to address any questions. And then on our website, you can also subscribe for our quarterly newsletter, which is the iConnect newsletter. And you can sign up on our website to do that if you want to be kept up to date. Obviously, later this quarter, we are looking forward to the readout of our Illuminate trial. So around that time, I'm sure we'll be in touch with Hope and Focus again and provide you with some information that can be disseminated throughout the community as well. We're very much looking forward to that. And for now, I want to thank you, Roseanne, for having me on the webcast.
Well, you're quite welcome. I do have one other question that
Sure, go ahead.
A viewer wrote in, will you be working on any other LCA genes? I know you mentioned it's part of it is where the science brings you, but any work on other genes?
Yeah, we are working on other genes, also other genes in LCA. There's many that we work on in the lab, so I don't know all of them from the top of my head, but yeah, there is a broad interest in IRDs, and LCA is obviously a disease that's really close to us that we would like to make a big impact. So yes, we are looking at other LCA mutations as well.
So you've got some of that in preclinical work? Correct.
Yeah.
Okay. All right. Okie dokie. Let's see. I think that's it. Daniel, it has been a pleasure to have you be part of our series, and I hope that our viewers enjoyed this as much as we did. And we just thank you for participating, and we look forward to hearing your good news.
Same here. Thank you for having me, Roseanne.
Okay, you take care.
Thanks, you too. Bye-bye.
Okay, bye-bye.